only chose 3 days with the most observable, um, with the most observable differences in the results. So, the first growth of mold in the experiment was observed on the third day. Small molds started to grow in the replicates of wet sunlight setup. A yellow stain in the bread also started to form in the wet dirt setup. Meanwhile, no signs of mold were observed in the dry setup. The color of the mold is black and the texture is fuzzy. So this picture shows the first picture shows the yellow stain on the wet dirt setup, and the second picture shows the mold on the wet light setup. So on the fifth day, more and bigger molds formed in the wet bread set of both setups. However, there are more molds that form in the wet setup exposed to light than the setup placed in a cabinet. There are already clusters of small molds that are growing inside of the dry bed of both setups. The color of the molds are black and green, and the texture are fuzzy and smooth. So on the eighth day, or the last day, almost all part of the surface of the bread of the wet light and wetter setup is already covered with molds. There are more types of mold that proliferated in the wet dark setup, which now also includes a light yellow type of mold. Meanwhile, in the wet light setup, the breads are mostly covered black molds. In both the dry light and dry wet setups, a few more molds form the size of the bread, while there are only a few molds on the surface of the bread. The color of the molds are black, white, yellow, and green. The texture are fuzzy, smooth, and lumpy. So the picture on the right shows the differences between the two setups of the differences between the dry and the wet setup. Final observations. From what I have observed, the wet bread setups in both light and dark areas are the first to grow molds, while the dry setups have grown a little to no molds 5 days later. The wet light setup is the first to grow molds, but the molds in the wet dark setup grow faster and more types of molds are also observed. The wet bread in both the light and dark setup have been almost completely covered with molds after 7 days. Different types of molds have grown and proliferated in both setups. Meanwhile, the dry setups of both the light and dark only a few molds have grown after several days. There are certain conditions that a mold needs in order to grow and proliferate. Molds grow rapidly when exposed to moisture, oxygen, food or organic matter, and warmth. When all these are present in an environment, then mold is likely to grow in that area. That would be all for Thank you.
Okay, thank you for that. You have, what's this? Comparing the results is on the dark and nakita mo kung paano nag-spread ng husto yung iyong species dun sa lalagyan mo at dun sa bread mo. Okay, can you give me 5 minutes, everybody? Ang susunod si Carmela, si Mark, and then si Meryl. Pero give me 5 minutes muna. Titingnan ko lang nanay ko. Ha?
Yes po, madam.
So, Christine?
Yes po, ma'am. Yes po. Ay, tapos na po ako, ma'am.
Ay, wait lang.
Ako po yung last.
Ay, okay. Sorry. Carmela and Mark, and then Meryl.
Good afternoon po, ma'am.
Opo.
Nakikita na po ba?
Yes po.
Good afternoon, everyone. I will be presenting my results for the experiment. So based on the result, molds tend to grow in places with a lot of moisture and mostly in dark areas. My dry bread samples in the light area did not have visible molds on them, as well as the dry bread samples in the dark area. This indicates that moisture or dampness dampness plays a big role in the growth of molds. One wet bread sample in the light area grows a red and brown mold, the Neurospora crassa and Cladosporium, on it, while all of the wet bread samples on the dark area had grown a variety of mold. As you can see in the photos, there's a black mold which is the Rhizostolonifer, one of the most common bread mold. Penicillium, the white-gray, Cladosporium, deep green to black, and the Neurospora crassa, red mold. So, overall, molds multiply by the release of small spores which are invisible to the human eye and float through both outdoor and indoor air. Molds can begin to grow indoors when mold spores settle on moist surfaces. Mold comes in a variety of forms and none of them will develop in the absence of water or moisture. Indoors, it is difficult to eliminate all mold and mold spores. Some mold spores will be present floating in the air and home dust. Mold spores cannot proliferate in the absence of moisture. Controlling moisture inside may and should help to avoid manage mold. That's all.
Okay, thank you for that. Meron kang mapula. Yun ang tatanong ko sana sa kahapon. Sabi ko, baka yung bread mo ay may mapula. But that's a beautiful observation. At yung meron kang nakuha na ganun. Thank you. Next is Mark.
Hello ma'am, good afternoon po.
Opo.
Ito po. Nakikita na po yung screen ko.
Yes po.
Okay na po. So,
Opo.
good afternoon po. So, here's my observation. So, the following images of bread mold labeled C1 and C2 means control group and will serve as reference to see the difference. DD2 stands for dark dry setup. This was the group that was dry and inside the cabinet, as well as SD2 stands for sunny dry. This also was the one exposed to sunlight. DW3, on the other hand, stands for dark wet setup. This was the group sprayed with water and put inside the cabinet, while SW3 also stands for sunny wet setup. This was the wet setup exposed to sunlight and sprayed with water. In the group that was exposed to sunlight, the wet bread seems to have a significant growth than with the dry bread. The SW3 has shown green. SW3 has shown the shown greenish gray growth with bumpy texture, while in the dry bread SD2 it has lesser mold and only one spot has severe mold growth. Then in the dark setup, DD2 and DW3, the one who has severe growth was the DW3 here. And it has a bumpy structure here like filaments, and colors varies from black, dark green to grayish yellow. On the other hand, DD2, so here's the DD2. On the other hand, DD2 also has mold growth but not as severe as the DW3. The color was more on ash gray with tiny filaments to dark green. And the control group serves as the reference on how mold grows and changes its color throughout the experiment. So I think Rhizopus and Aspergillus ang mga nag-grow po sa bread ko. Then the colors has been final observation, the colors has been changing throughout the entire experiment. There's difference in color in each day of experiment, and there's significant differences between the breads in dark areas. In dark areas, mold has been fast growing for they need moisture to grow, while in the lighted areas, they tend to grow slower. Same happened to wet bread. It grows faster than in the dry bread setup. This knowledge from bread mold experiment can be used to avoid food contamination at home. I can use this knowledge to prevent mold growth and to save food from fast contamination of molds. Then, much like any other type of living organism, mold requires a source of moisture to sustain itself in addition to food for nutrients. However, in the other organism, mold is incapable of getting up and moving between a food source and a water source. Thank you po.
Okay, thank you for that. Baka malito ka sa mga malito ka sa mga, ang tawag dito? Malito ka sa mga codes mo. Sa report mo na final report, lalagyan mo yung ano mo ng listahan ang mga codes mo ha, kasi
Okay po.
baka malito-lito ka. Okay, Meryl and Miel and Sin Ira.
Good afternoon po, madam.
Yes po. Eve.
Yes po. Nagpe-present na po.
Yes po.
Oh. Okay lang. Ay, thank you kagad. Wait lang po. Minor ano lang po. Minor difficulties. Okay po. Good afternoon. I will now be sharing my observations for our bread mold experiment.
Po.
Okay, first I'll talk about the setup exposed to light, which is set near an open window.
Wala po kami nakikita.
Here, ay wala po.
Wala po, dark. Dark po.
Pwede po next na muna. Ano ko lang.
Yeah, si Miel muna.
Ayusin ko lang.
Miel?
Opo, thank you po.
Oh, yes po.
Opo.
Alis po. No, person.
Eve, patanggal ng presentation mo.
Opo.
Wait lang. Nakikita na po lang yung presentation.
Yes po.
So, good day everyone. I'm Yal Aimikos. Allow me to share my results regarding the week-long activity and bread mold experiment. So, here is the summarized data of my observation from day 1 until day 7 of the experiment. As in the table, no mold was observed during the first day of observation, while molds grew on the succeeding days. On the second day, black and white mold started to grow on wet breads for both light and dark setups. On days 3 and 4, the mold grew more for both setups. Green and black molds start to show on day 3 and for both dark and light setups, while an additional white mold is observed only for dark setup. Brown molds can be seen on day 4 of both dark and light experiment. On day 6, yellow molds are only present on dark setups. So yellow molds are present here only on dark setups, and majority of molds in dry setup has smooth and fuzzy textures, while fuzzy and lumpy textures are observed in wet breads. So in terms of growth po, up until the 7th day, wet breads grew more in height and in number than dry breads for both dark and light experiment. So this is just to show the summary of the results. So as seen po in the experiment, molds grew best in warm conditions, hence the sealed containers allow the growth of molds. Also, mold usually grows in both light and dark conditions. However, molds usually favor growth in dark areas. In contrast to plants, molds don't require photosynthesis and do not use light to obtain energy. As a matter of fact, sunlight can impede mold growth, and in some cases, this kill molds. So molds prefer darkness to grow. And in addition, another requirement for mold growth
is moisture. Molds thrive in wet and humid conditions to allow them to grow and spread. So this is evident on the results of the experiment since majority of the molds are found in wet beds under dark conditions. So experiment is vital in our understanding of mold growths, as well as increasing our knowledge in the prevention of mold growths. So since we know that mold growths that grew best in warm, dark, and humid areas, we can apply this learning in minimizing mold growth in indoors. So by keeping areas well ventilated, clean and dry, we can reduce mold growth. That would be all. Thank you for listening.
Thank you for that. And yung pare-parehas kayo na on the first day and on the second day halos wala pang nag-grow. But on the third day nakikita nyo na. And paminsan mga dot-dot lang. Yung hindi nyo pa nakakita kasi yung pag-spread nung hyphae. So ang nakikita nyo pa lang ay just a small portion of the hyphae. But eventually, nung nakita nyo na at lumabas na yung mga fuzzy bodies na nakikita nyo and thread-like structures, yun, nakapag-spread na at nag-grow na yung inyong hyphae. Okay, thank you for that. Next one is Miel.
Ako pa yun, ma'am.
Ay, sorry na. Oo na. Wait lang. Ay, ano ba itong listahan ko? Wait lang. Yeah. Sin, Ira, and Sofia, and Giancarlo. And Dinah, and Daryl. Wala si Dwayne. Nilayasan tayo. Sino pa ba ang hindi ko natawag? Sige, ikaw na, anak.
Good afternoon, Madam. Permission to share script po.
Yes po.
So before po I start my presentation, I'd like to apologize for the background voices. Batay po kasi ako sa shop and I can't go home for the meantime. So moving on, in this experiment, mold growth were observed in all wet samples in both dark and light areas. Dry samples, on the other hand, were found to be mold-free. As a result, only the wet samples are shown in this presentation. This slide displays the fourth day of the experiment and the first day to observe mold growth. As you can see, all of the wet samples in both dark and light areas were observed to have growing molds except in light area 2, where the sample is only observed with inverted lumps indicating dryness. The following slide reveals the 8th day of the experiment. In this, all of the samples have been colonized by colorful molds, namely white, green, and yellowish molds, with the exemption of light area 2, which is experiencing mold growth for the first time. Furthermore, the majority of the molds in the samples appears to be fuzzy and lumpy. So to conclude, molds break down the organic substance of the mold as soon as it started to spoil due to its dampness. From this, we can conclude that the mold requires 4 things to grow: water, food, oxygen, and an area with a temperature that it can survive. Moreover, keeping the food dry and properly sealed reduces the chances of food spoilage, thus lessen the likelihood of mold. That would be all po. Thank you.
Okay, thank you for that, Ira. Parehas din ang iyong mga and fuzzy, and later on, nag-form ng mga bilog-bilog, and nag-form ng, nag-change ang mga colors, and eventually, it's growing really well, and of course, with water, kasi yung walang water dry, wala silang magagamit for growth and development naman. Okay, Sofia, Giancarlo, Dinah, and Daryl ang last. So, sino ang hindi natawag?
Ako pa po, madam.
Ayan, Frances. Sige, ikaw ang last, Franz.
Okay po, madam.
Okay.
Good afternoon
Sige.
po.
Pia.
Share screen lang po.
Yes po.
Sorry po. Nakikita na po. Oh my God.
Yes po. Oh my God. Nakikita po.
Okay. Good afternoon, everyone. I'm Sofia Hanarikerke, and I will be presenting the results of my bread mold experiment. So the first one, for the breads that were um, exposed to light, there were 3 wet and 3 dry. The wet ones started showing visible changes on day 2, and then more types of molds grew after a few days. So
Wow.
pinakamarami and pinakakita po yung color white with fuzzy textures, and more molds with color dark green, yellow, brown, black, and green are also visible. As yan po sa pictures. And then for the dry breads po, the mold started to grow on day 4, and yung kita lang po doon nung day 4 is yung fuzzy white ones, tapos barely seen pa po siya. And then no few days po after, lumabas na din po yung color white and then black. Yung white po meron din po siyang fuzzy, meron po siyang both fuzzy and lumpy one. And then for the breads that were kept inside the cabinet, there were also 3 wet and 3 dry. Then the wet ones started growing on day 2, yung color brown po. Tapos parang smooth siya or parang stain lang po siya dun sa tinapay. And then molds continued to grow after a few days, which were color white, dark green, green, black, yellow, and brown. Yung white po, as you can see, dito po sa third picture, yung sa wet po, na-colonize niya po yung half ng cup, and na-adapt po nung white na yun yung shape ng bottom ng cup. And then,
Teka lang. Bago ka magpatuloy, is that cup covered? Did you grow
Yes
that with
po.
cover?
yes po.
Na hindi exposed? Kung hindi, mai-spread mo sa bahay mo ang ano. Did you open it? Yes, you opened it.
Yes, I opened it po. Kanina
Sabi ko
pa lang.
baga, no, I said do not open. Diba? Hindi ko pa sinabing i-open. Nag-open ka na. And then, why in a cup? I said in a plastic container para flat because there is an effect on the container. Dapat yung bread mo is flat. You did not follow my instructions. Sige, patuloy.
Sorry po. Then, for the dry breads po, it started showing molds on day 5 na po. Then, white po yun and lumpy. Sa gilid lang po siya. And then, the next 2 days, yung color green and yellow po, nag-grow na din. Yung yellow po, same sa kanina, parang stain lang din po siya. Then, yun lang po for my final observations. Dark and wet setups was the fastest to grow, and yun din po yung pinakamarami. And then second po yung light and wet setup. Then mas madami po yung sa dark and dry setup compared sa light and dry. Then yun lang po. Thank you po.
Okay, sige. Next time, follow instructions. Carlo?
Yes po. Yung akin po ma'am, nag-ulit po ako kahapon kasi yung setup ko po, naulanan po, nabasa po lahat. Pero i-present ko lang po yung naisalba kong ano.
Di ba merong takip, Ib? Nagtataas ka ng kamay. Ib?
Present ko na lang po ma'am yung
Hello
naisalba.
po, madam.
Nagtataas ka ng kamay. Wait lang, Carlo. Ib, nagtataas ka ng kamay.
Opo. Nakababa
Yes
na
po.
po. Can I have a second attempt po sa pag-present?
Bakit?
Hindi pa po ako nakapresent.
Pinawag kita kanina eh.
Opo, nag-ano po siya, nagsabi po kayo.
Ib. Opo, babalikan ko ikaw. Yes po. Okay.
Sige po.
Sige, si Carlo muna. Carlo?
Sige po.
Carlo?
Ang
Teka lang, si Carlo.
ay,
Teka, Carlo. Di ba may takip? Ba't naulanan?
ano po ma'am, manipis po kasi yung ano, yung pagkalagay ko po ng plastic. Tapos malakas po yung tulo kasi bumagsak po talaga ma'am yung ulan sa amin. Tapos yung
Ay,
yung
di ba kasi plastic container? Anong plastic ang nilagay mo?
sa disposable po na plastic cups lang po.
Naman, tayo. Sabi ko container, hindi cup. Di ba? Yun. Sige, Carlo. Balikan ka namin sa Biyernes. And you
Sige
can
po.
repeat, but you have to use a flat container,
Sure
not a cup. Okay, sige. Babalikan ka namin sa Biyernes bago tayo mag-lesson. Ikaw muna.
Sige po, ma'am. Thank you po.
Sige. Balikan. Teka lang at ililista ko. Si Dinah muna, si Daryl, si Franz, at si Eve.
Ma'am, nag-report na po ako kanina.
Wala.
Oh, bago po ni Donna.
Yes. Sorry na, sorry. Daryl. Oo nga pala. After ni Corn. Daryl?
Yes,
Si Daryl, Franz, at si Eve.
wait lang po. Share screen na po.
Itong listahan ko na to at nagduduro-dubli. Kasi na-leave group ka.
Nawalan po kanina naman.
Sige. Okay lang yan. Sorry na, matanda na rin ang lola niyo dito. Tao lang.
Kita po.
Yes po.
Good day, everyone. I will be presenting the results of my bread experiment. Experiment po, madam, only lasted 45 days po since I started.
Hindi ka pa rin tapos?
Ay, hindi, 5 days po. Kahapon po.
Kahapon ka nag 5 days?
Ang ngayon po, madam, yung
Teka. Sandali, hindi kita naiintindihan. Kahapon ka nag 5 days?
hindi, kahapon po. Yung ika-4th day ko po, madam.
Sige, balikan ka rin muna namin sa Monday. Ay, sa Friday. Okay, Okay. Sige, Franz? Franz?
Yes po, madam. Good afternoon po.
Una, and then si Eve.
Madam, okay lang po ba na hanggang day 6 pa lang po ako?
O, hindi ka rin. Sige, balikan ka rin namin sa Monday. Si Dada,
Okay
si
po.
Franz. Si Eve na ang last. Balikan ka namin sa Friday. Plus si Dwayne. Pasabi kay Dwayne na mag-attend ng klase. Hindi yung sa una lang, alauna, o pagsimula doon lang. Palagi ginagawa. Sige, Eve.
Opo, good afternoon po ulit.
Yes po.
Naka-present na po ba?
Yes po.
Good afternoon po ulit. This is my second attempt. I'll talk about the first setup, which is exposed to light. So here po, you can see the before and after photos of the setup. So it's a comparison between day 2 and day 9 observations. I opted to show day 2 because this is when I first observed growth in some replicates. The left side has the dry replicates, while the right side has the wet replicates with 3 to 4 sprays of water each. By the second day, the dry replicates had no visible fungi growth, but as days passed, it steadily grew green mold, observed here, which has both smooth and fluffy textures, and some brown mold, which is seen here, which is lumpy. And in the wet replicates, there was already a visible growth, which is seen po sa gilid, sa crust po ng sliced bread, which first appeared flat. By day 9, green, brown, and black mold have already grown and spread, and their textures are lumpy, smooth, and fluffy. The next po is the dark setup, which is contained in a cabinet. Right off the bat, you can see a clear difference from the setup exposed to light. Similar to what I previously said, the dry replicates kept in the dark also showed no visible signs during the second day, but it eventually grew green, brown, and white, and some yellow mold, which collectively have lumpy, smooth, and fluffy textures. For the wet replicates, second day na rin po siya nag-show ng signs of mold growth sa gilid din po, sa crust. So, by the ninth day po, the mold, the colors of the mold are green, yellow, white, black, and their textures are fluffy, smooth, and lumpy.
If
Apo, yes
anong
po.
lalagyan mo yan? Ano yung container mo?
Ano po siya, plastic plate, but ang ano po, ang pang-seal ko po is plastic wrap, saran wrap po.
So, nakadikit sa bread mo?
Ay hindi po,
Hindi
Parang
nakadikit
po.
ang saran wrap sa bread?
hindi po. Ang
Okay.
ang white po ano po, flash.
Sige.
Nag-reflect lang po. So yun po, regarding po sa spores, I first observed their presence around day 4 because when I move around the bread, it would leave some residue. Okay, so
Impakan mo?
ay hindi po, nagsislide lang po sa container.
Yung alog-alog? Okay.
Opo. So yun po. This is the fine ang summary. So the least growth was observed on the dry replicates exposed to sunlight, while the most was observed on the wet replicates kept in the dark. So this indicates that the mold best thrives in dark and damp conditions. Yun lang po. Thank you.
Okay, thank you for that, Eve. So hanggang kay Eve tayo, wala si Dwayne, and there are several people na hindi nagpasok kasi walang may brown out. Yung iba naman ay, ano man tawag dito? Ay hindi malakas ang mga signal. But anyway, just to reiterate on instructions, you have to follow instructions. The reason why I ask you to use a disposable container na flat instead of asking you to use it on a cup. Ang iba, actually, ang ginagamit nila ay just the ziplock. Yung plastic lang talaga doon lang inilalagay. So, ang purpose ko is for you to be able to observe nga talaga yung tawag dito, yung bread na nag-grow na dire-diretso yung kanyang hyphal bodies na pag-grow. Para yung matting na nakikita nyo is already the extent of the growth. Nakita nyo naman kung paano nag-extend yung kanilang mga growth. But when you use a cup na twist din yung mga growth ng mga hyphal bodies at hindi siya basta-basta nag-i-spread ng husto at hindi mo ma-observe yung buong bread. And also for observing also anong nangyari doon sa water. Earlier, somebody said na naging cloudy na yung kanyang naging cloudy at may tubig kaya hindi niya makuhanan ng maayos na picture yung kanyang setup. It is because yung water na nilagay mo doon sa bread ay nag-evaporate din, but hindi siya nakakalabas of course doon sa plastic container mo because it is closed. So in some cases, there are still many factors that actually affect the growth and development of your fungal bodies. At yung nakikita nyo na mga lumpy things na may mga bilog-bilog, these are the fruiting bodies, and inside the fruiting bodies are actually the spore. And with the case of Eve na nagagalaw niya, na-shake niya, so makikita mo dun kapag nag-i-spread yung spores. So, please post these presentations on the area where I placed in Google Classroom. Ilagay po itong ngayong araw na ginawa. Just the ones that you presented. Hindi na po kailangan ng explanation. I just want you to put things there. Nilagyan ko hanggang alas 5 ng hapon ang paglalagay kasi of course mahina ang mga signal. Baka mamaya hindi basta-basta mai-post. And then I also placed already the new material, fungi characteristics and fungal characters and roles na lesson para sa susunod na Biyernes. But bago tayo mag-lesson, babalikan natin yung mga uulit. Si Carlo uulit at yung hindi pa nakatapos ng 7 araw. Ang iba naman ngayon actually ay 10 araw na. So today for the final picture of your outputs, I want you to open your setups. Pero naka-mask. And I want you to open your setup outside of your house and take pictures of your setup as a whole and take specific pictures picture of specific features na makikita nyo doon sa inyong mga setup. Kasi you have to remember, iba-ibang klase ang kanilang mga fruiting bodies. So by opening, you are actually looking at the fruiting bodies. Lahat naman meron na at iba-iba ang itsura ng kanilang mga fruiting bodies. So I want you to zoom in, something like that, on the fruiting bodies at kung ano na ang structure ng kanilang mga hypha. Intindihan po ako ninyo. And after that, you have to discard your setups. You have to throw away your bread, preferably ililibing. Ibabaon sa lupa yung inyong bread sample. At hindi basta-basta ito-throw out sa basurahan ninyo sa loob ng bahay. Kung ito-throw out na sa basurahan, ilalagay pa rin po sa plastic na hindi po natatalian yung plastic at ibubuhol yung plastic para hindi po tayo nakapag-spread ng spores doon sa loob ng inyong bahay because it can contaminate everything sa loob ng bahay. Okay po, naiintindihan po ako.
Yes po, ma'am.
Tanggalin mo nga yung tanggalin mo ang presentation mo. Wait lang, Daryl.
Hello po, madam.
Ay, hindi. Mag-o-open ka sa 7th or 8th day mo.
Okay po, madam. Noted po. Thank you po.
Pero si Carlo ay magsisimula sa simula and Carlo, you have enough days hanggang 7 days. Hanggang 10th day nga yata.
Sige po.
And you are going to open up afterwards and then discard. So please follow my instructions kung paano tayo mag-discard ng ating mga samples dahil makakapag-contaminate tayo sa loob ng bahay. Okay, so makikita nyo doon, I posted already on your free time. You have to check the PowerPoint presentation and if you have more time to browse on the internet for some other information and read from the books that I give you, please do. At ano rin po tayo, mag-i-schedule din po tayo ng makeup class based on the based on the memorandum that was given by the President to do makeup class para dun sa isang linggo na hindi tayo nagklase. But hindi ko sinabi na synchronous ang ating makeup class. Pwede namang asynchronous ang ating makeup class. Kailangan ko lang tingnan ang common schedule para sa inyong lahat dahil meron pa kayong iba-ibang subjects na hindi kayo magkakasakasabay. Okay po? So, do you have questions for this afternoon? Questions? Meron po bang tanong? Ang submission po ng final report will be until Sunday. Yes, Paul.
Yung paglibing po, ma'am, ng breads, di man po makaka-apekto sa mga surrounding plants.
Hindi naman po, Paul.
Ay, okay.
Because it's naturally occurring in the soil.
Okay po, ma'am.
Okay po. Naturally occurring siya sa soil, sa totoo lang. Kaya hindi po siya makakapag-contaminate. It will, hindi naman siya basta-basta mag-grow doon sa soil because it's another medium. But it is naturally occurring in the soil. Okay po? Pero kung ayaw nyo, then pwede namang mahirap kasi pag susunugin at mahirap na ilalagay sa microwave, mako-contaminate ang microwave niyo. Okay po? So the best thing to do is to actually just ilibing na lang at ibaon na lang sa lupa ang inyong samples. Or i-throw na lang sa basurahan basta basta nakasil ang plastic na hindi makakapag-contaminate. Okay po? Sige. Somebody was saying, Sofia?
Ma'am, excuse me po. Tanungin ko lang po kung uulitin ko pa po yung nasa cup po kanina.
Ay, hindi na, Sophie, kasi nagawa mo na. Sinasabi ko lang sa susunod, pag-follow ng instructions. Iyo po.
Okay po. Thank
Ang
you
dami
po.
naman ating mga lalagyan na pwede yung mga pinagpo-food panda nyo nakalagay dun sa mga lalagyan na ganon and everything. Okay? Sa susunod, magsusunod
Opo,
para
sorry
mas
po.
maganda ang iyong results. You don't have to be sorry. It's okay. Sige, if wala na pong tanong, I will say goodbye to everybody. Thank you for sharing your results and thank you for doing the lab activity na kayo-kayo lang. Dahil kung nasa loob tayo ng classroom, ay sabay-sabay natin itong ginagawa. Okay, so bye everybody. I will see you again on Friday. Thank you, and then
Thank you, ma'am.
we can always chat. Bye!
Thank you po.
Bye, madam. Thank you po.
Thank you po.
Thank you po.
Thank you po, ma'am.